Ein König ist der Wein, ein König ist der Wein, mit Segen reich beladen, ist er von Gottes Gnaden und mancher Wohnung sein. Ein König ist der Wein, ein König ist der Wein. Ist der Wein in seinem grünen Lande und schwingt er alle Lande, beherrscht sie groß und klein. Ein König, ein König, ein König ist der Wein. Ein König ist der Wein.
So as I mentioned when we started off, I said this is a tradition for 15 years. And I think when we started together with Science and AAAS, I think the whole purpose of it was to create a prize that would motivate young scientists. There are a lot of awards, but I mean, not really for young scientists. And why did we want to do that? Well, there are a couple of reasons. Number one, I think it's very important for all of us that science is progressing, and to make young people interested and motivated in science is very important to society and to progress in the world. And particularly now when there's so many tough challenges ahead of us. No further ado, I would like to invite John Travis to introduce the prize winners and let the ceremonies begin. John, give him a warm hand.
Well, I first want to thank Peter and GE Healthcare and Life Sciences for being such a long-term sponsor of this. Science is a pursuit that involves a lot of passion and dedication and isn't an easy profession. It's often one in which rewards are few and far between. Because in 2005, Science celebrated its 125th anniversary, and we did a special issue where we talked about the 125 biggest questions in science. And one of those questions was, how do organs and tissues know their right size? How do they know when to stop growing? How does a mouse heart stay small versus an elephant's heart grow much bigger? And to a certain extent, that's what Michael worked on.
What a great introduction. As a matter of fact, when I was submitting the paper to Science, I actually reminded them that they considered this to be one of the 125 most interesting questions. In science, you learn to relish the good times because, as was alluded to earlier, they don't come around very often. So thanks everybody for being here tonight and helping us celebrate the good times that the four of us had during graduate school. This award would be amazing for anyone to win, of course, but I'm sure that any of my college professors would tell you that for me it's particularly amazing. I was a terrible student at university. I didn't go to class very often, I didn't study. In fact, it became so bad that one day one of my professors pulled me aside, told me that I was dumber than a bag of hammers, and that I should switch to an easier major. We really tried to solve mysteries. And for us, I think the biggest mysteries for the future lie in the brain. This is the question Dragan and I will be trying to ask for the foreseeable future. I'd love to hear everybody's thoughts about it as we continue to enjoy this really extraordinary night. Thank you, everybody. Thank you, GE Healthcare and Science. This is magnificent.
First of all, I would like to thank the organizers of GE in Science, as well as the judges for this great honor of being selected as the regional winner for Europe, and also, of course, for the invitation to come here to Stockholm. So, an amazing night so far. My PhD studies conducted at Harvard Medical School focused on one central part of the so-called innate immune system against viral infections, and this is the well-known interferon system. Specifically, my work focused on the mechanism how actually our cells detect viral infections, and also on the signaling cascades which then finally lead to interferon production to limit the viral replication and also the spread of the viral infections. Thank you very much for your attention.
The ideas of these molecular regulations were derived from the curious results obtained in this live cell protein activity imaging experiments. So the live cell imaging like this should be beneficial for understanding of the mechanisms, the molecular mechanisms, molecular functions regulated spatially and temporally in living organisms. And that's my thesis study. Thank you so much for your kind attention.
So almost a decade ago, in 2001, the first draft of the human genome was sequenced. And in my research, I'm trying to figure out how to read this blueprint of life, as we call it. And in particular, I'm interested in understanding how it is that even though each of the trillions of cells in our bodies carries the exact same DNA sequences, they manifest tremendous diversity in their structure, function, and in response to external stimuli. Thank you.